Today we are very lucky to have Dr. Nurul Izzah Muhammad Sarmin to share her research interests and her experience in doing her very exciting research so far. So now a few introduction about Dr. Nurul Izzah. Dr. Nurul Izzah is actually our research fellow in Ataur Rahman Institute for Natural Product Discovery. She is also a senior lecturer in microbiology of Faculty of Dentistry, UITM. And her research interest is about plant endophyte interaction and all the metabolites derived from the plant endophyte relationship. So without further ado, Dr. Nurul Izzah will present her exciting work.
Yeah, Assalamualaikum and very good morning. Today I want to share my part of my research interest, which is endophytic microorganism. So this is like a sharing session that what I've done in my in Ataur Rahman Research Institute for Natural Product Discovery. Okay, as introduction, what is microbial endophytes? Which is this endophyte has been introduced by De Berry in 1866, where this endophyte is defined as any microorganism which grow within the plant tissue. So any microorganism which grow within the plant tissue we can call as endophytes. And more precisely, they can be described by their types, whether it is fungal or bacteria, and their relationship to the host plants, whether they are obligate or facultative endophytes. There are also comprehensive definition of endophytes where they will not specify the functional relationship, and apart from the commensalistic symbiont, they can exist from latent pathogens or saprophytes to mutualistic associations. So how they get the mutualistic association? Actually, this endophyte is a microorganism which reside inside the tissue plant, which without causing any overt effect to their host plants. So it means that they have their mutualistic association, so they have win-win situation. Yeah, the plant endophytes relationship affect the survival and health of the plant very much. So means that they can improve the health of the plant. And for example, the rhizobia legum symbiosis, which is the bacterial endosymbiont, they will govern the plant's need for nitrogens. The endophytes also will induce the host plant to produce metabolites that promote the plant to grow and help them to better adapt to the environment. Endophytes play an imperative role to maintain the health of the plants, and they can protect or prepare the plant against abiotic and also biotic stress, and helps in enhancing growth and the yields. They help in the biotic stress, whereby they help their host plants to protect from other microorganisms, other pathogenic microorganisms. And for the abiotic stress, they protect their host plants in preparation to the salinity or any other metal, organic metals, and also other environmental stress. Okay, how they the lifestyle of the endophytes, how they can get a mutualistic agreement with their host plants by producing the flavonoids. So the flavonoids is actually the root of the plant is very rich in biomolecules, and also the exudates of the plant also rich in nutrients and also water. So all of these, they can attract all sorts of microorganisms. But the microorganism also will also filter whatever comes, whether they are pathogenic or non-pathogenic one. So actually, they talk each other. So they can produce signals to cross-talk each other. So this one will attract the endophytes to come to the root of the plants, and also when the host plant recognize whether this one is non-pathogenic one, so they can allow the endophytes to get into the host plant's tissues. And one of the chemoattractant that has been released by the host plants, for example, is the flavonoids. So the flavonoids are one of the metabolites secreted by several plants, and also play an important role in the endophytic interaction with the root hair. In the presence of these metabolites, the colonization of root in rice and wheat by endophytic Ceratiae, EDA and Azorhibium caulidonans was more effective compared to without the presence of the flavonoids. And the plants also has lipochaetoligosaccharides. So this is signals where they can these signals will be activated during the symbiotic pathway. For example, the strigolactone secreted by the roots of Arabidopsis thaliana was found to act as a signal molecules for colonization of the endophytic mucor species, which is endophytic fungi. And another example, which is the arabinogalactan proteins, which is AGPs, which is highly glycosylated members of hydroxyproline-rich glycoprotein, superfamily of plant cell wall proteins. This one also will attract, they will produce establishing interaction between plant and also the microorganism, including the endophytes at the several stages of the life. So the plant also has the arabinogalactan proteins, which is AGPs. So this one is highly glycosylated members of HRGP superfamily of the plant tissue wall proteins. So they can establish interaction of plant with microbe at the several stages. And these proteins also have a definite role in root colonization. They're working as repellents or attractants for microbes in development of infection structures. So when with these AGPs, so if the microorganism comes at the root for colonization is a pathogenic microorganism, so they are able to repel the microorganisms. So other compounds which involve in the mutualistic agreement is including the sugar, amino acids, and also organic acids, phenolic compounds, and other secondary metabolites, which secreted by the plant roots, which is they can selectively invite the mutualistic microbes, particularly the endophytes. So how they recognize whether that one is endophyte or pathogens? So they have a receptor, which is this the receptor kinase, and also this receptor kinase able to recognize whether that one is diaphragms, or whether it is a bacterial endophytes or also fungal endophytes, or the second line is the is also a kinase. So they can recognize whether that one is bacterial or fungal endophytes, and then this one will activate the plant defensins, PR protein, also phytoalexins pathway in the host plant's defense response. So how there is downregulation of the plant defense pathway during colonization of plant by mutualistic partner, such as the rhizobia or arbuscular mycorrhizal fungi. During mutualistic interaction, late induction of ethylene or jasmonic acids and also salicylic acid signaling pathway will prevent the microbe from overstepping and overpowering the plant. So they have the mutualistic agreement. And the plants via nutrient monitoring are able to identify whether the invading microbe is beneficial or a pathogenic microorganisms. So the lateral gene transfer in the root of the host plants is they have also the gene for mannitol dehydrogenase in several bacterial endophytes, and also enzyme which involved in the defense against phytopathogenic fungi. And however, not a single factor or mechanism can be ascertained for the endophytic lifestyle, because there are so many things that yet to be discovered for the for the host interaction between the endophyte and also the host plants. So the endophytes also able to modulate the plant's immune system for them to able to have to make a mutualistic agreement. So they have a recognition of conserved molecules, which is the characteristic of the many microbes. For example, the microbe-associated molecular patterns or pathogen-associated molecular patterns by plants. So they have these conserved molecules that is able to, for the host plant to recognize whether that one is endophytes or also or pathogenic microorganisms. So the MEMS are recognized on the surface of the plant cells by pattern recognition receptors. And for the fungal endophytes, where the fungal endophytes has the PR3, which is the chitin specific receptors, on the plants will recognize the chitin oligomers formed on the fungal cell wall. So this one, they will able to recognize whether that one is fungal endophytes. However, the endophytes also work out mechanism to protect themselves from plant defense mechanism. For example, the fungal endophytes will produce the chitin deacetylase. So this chitin deacetylase will deacetylate the chitosan oligomers that are not perceived by plant's receptor. Hence, they will be hence this deacetylase will prevent the fungal endophyte from being recognized by the host plants. The endophytic bacteria are also known to produce their own MEMS, which are not recognized by the PRR of the plants, or plants trigger a comparatively weak or transient defense reaction compared to the pathogenic interactions. So they will also able to prevent themselves from defense mechanism of the host plants. Protein secretion system in the bacteria also will modulate the plant immune system, which they are either absent or present in the low evidence in mutualistic endophytic bacteria. So this is the example, this illustration of how the fungal endophytes produce the chitin deacetylase. So they will able to be recognized by the host plant. So this is how they able to prevent themselves from being recognized by the host plants. So the endophytes also will increase the resistance to biotic stress during their mutualistic agreement. So the potential of endophyte will suppress the phytopathogens via the antagonistic activity. This one, it means that this endophyte will help the host tissue to being suppressed by other pathogens. And the endophytes are also known as to play roles in inducing systemic resistance against phytopathogens in plants. So they help the host plants to become more healthy. And the endophytic bacteria are also known to produce various volatile organic compounds, which is the VOCs, with a broad spectrum of antimicrobial activities against pathogenic bacteria, fungi, and also nematodes. So what are the plants are also known to produce low molecular weight antimicrobial molecules, for example, the phytoalexins. So these phytoalexins includes various metabolites of group of metabolites, for example, the terpenoids. This enhancement of terpenoids and also growth of the Euphorbia picanensis by endophytic fungi Fusarium species. There are also comparative studies by several workers showed that the endophytes containing plants report relatively high cellulose content, also laminar density. So because of the high cellulose content, also high in laminar density, so the plant leaf become more tough, and also will reduce the herbivory rates specifically by leaf cutting ends. The presence of endophyte in the host tissue can thus enhance their resistance against pathogens by eliciting the host response or by producing antagonistic metabolites themselves. So the role of endophytes in the production of plant secondary metabolites. So how actually this endophyte will help the host tissue to produce the secondary metabolites by their coexistence in the host plant tissues. The coexistence and evolution of endophytes along with their host plants has established a special relationship, significantly influencing the production of bioactive metabolites in the plants. In the communication of endophytic communities with the host plants will significantly influence the physiological process of the plant. For example, the host plant will able to activate the silent gene cluster, which leading to the synthesis of novel secondary metabolites. Both of the plant and also endophytes can also produce an array of common secondary metabolites from similar precursors, means the endophytes and also the host plants will can able to produce similar bioactive metabolites. The such bioactive compound, for example, the antibiotics, and also antitumor bisindol alkaloids, anti-cancer drug, for example, the camptothecin, podophylotoxins, and also insecticide azadirachtin from Azadirachta plant. The bioactive compound with anti-cancer activity is the synthesis of taxol from the yew plant and from its fungal endophyte Taxomyces endorrhoeae. So when they study more deeply on this endophyte, the Taxomyces endorrhoeae, so they found that the genomes of this endophyte did not show significant sequence homology with the taxol biosynthetic genes from the Taxus plant, which indicates that the fungus might have independently developed the taxol biosynthesis pathway. The hypothesis by Howitz and also Sinclair states that the homologous genes cluster present in the plants and also microorganisms may get cross-activated by stress-induced molecules from plant hosts or endophytes under certain conditions. So the discovery of from the discovery of endophytes, later on they found that these endophytes are ubiquitous with rich biodiversity, and each individual plant actually is the host of one or more endophytes, and they are also this endophyte is a chemical synthesizer inside the plants, and they can synthesize bioactive compound that can be used by plants for defense against pathogens. And the endophytes also has a role to for the production of alkaloids, terpenoids, flavonoids, and also steroids and others. So the most study that has been done for these endophytes, actually the endophytes are the major group of microorganisms which produce the antibiotics. So the antibiotics is the most bioactive natural product which has been isolated by the endophytes. For example, the antibiotic penicillin has been produced by Penicillium, which are endophytes. So the antibiotic, for example, the actinomyces is also one of the most antibiotic producer. And for example, the Streptomyces species is also for now there are so many research there are so many research about the Streptomyces as the main antibiotic producers. For example, they are now developing antibiotics against methicillin-resistant Streptomyces, Staphylococcus aureus, and also other pathogenic microorganisms. The new metabolites were detected to not only have activity against human pathogenic fungi and also bacteria, but they can also they are not only can act as antibiotics, but they can be also has the properties to be a fungostatic to plant pathogenic fungi. For example, when Izzah found the coronomycins, so this is a complex novel peptide antibiotics which has activity against fungi and also human pathogens, for example, the Cryptococcus neoformans. But this coronomycin also able to have other activities other biological activities. For example, this coronomycin is also active against malaria parasites, for example, which is the Plasmodium falciparum, at an IC50 concentration of 9.0 nanogram per milliliter. Weber and friends also found that the novel antibiotic formol isolated from the fermentation of endophytic fungus, Phomopsis species, from the medicinal plant of the Erythrina crista. So this found, they characterized these antibiotics as a polypeptide lactone. In 2004, they found two new fusicocaindietapines, which is preconicins A and B, which it has antibacterial activities, and these metabolites has been isolated by bioassay guided fractionation from endophytic fungi of Pericornia, which collected from the small branches of the Taxus cuspidata. And another example, which is Coletotrichum species, which is isolated from the Artemisia annua, which is traditional Chinese herb medicine, that is well recognized for its synthesis for artemisinin that been used for as an antimalarial drugs. The endophytes also now has been studied for its properties as anti-cancer agents. For example, one of the most surprising findings of endophyte study is the isolation of taxol producing endophyte, which is Taxomyces andreniae. So this taxol has been approved by FDA as one of the most potent anti-cancer drugs, but the supply of this drug has been limited for the destructive collective of the U3 as the main source of the taxol. So in the earlier years, so they found that this taxol has an anti-cancer properties, but they have to cut so many U3s to get the taxol. But later on, when they found this endophyte Taxomyces andreniae, so they found that this Taxomyces andreniae has the properties to produce the taxol. So when they studied the gene cluster, so they have the Taxomyces andreniae has a different sets of genes compared to their host plants. So now they believe that when they did extensive studies about this Taxomyces andreniae, so they found that this Taxomyces andreniae is the one that produced the taxol. It's not from the U3, so means that it's very interesting findings where the Taxomyces andreniae is actually the one who produced the taxol. So it means that they no need to cut more trees of the U3. So the endophytic fungus strain of the BT2 from the Taxus chinensis varmariae has been isolated and also was detected to produce taxol and its precursors, and the extracts of the endophytic cultures was shown to have strong toxicity to liver cancer cells 7402 and also lung cancer cells A549. Another example, which is the camptothecin, which is naturally occurring enantiomer, which was first isolated by the Wall et al. From the wood of Camptothecin acuminate, which is a plant native to the mainland China. This camptothecin is a derivative which shows strong anti-neoplastic activities, and this drug is also used in China for the treatment of the skin disease. So when they found the fungal endophyte isolate the camptothecin, so this endophyte has been isolated from the inner bark of the plant of Notapdophytes from the western coast of India. So they found this endophyte able to produce the camptothecin to treat as a strong anti-neoplastic activity. And another example is the fungus produce the anti-cancer drug, which lead the compound camptothecin. The biological activity of this compound was tested in vitro for cytotoxicity assay against human cancer cell line. So they possess the anti-neoplastic activity against lung cancer cell line, and also liver cancer, and also ovarian cancer. So these endophytes also has other biological biological control, can be as biological control agents. So this endophyte has been also used to help in the loss of biodiversity, in environmental degradations, spoilage of land or water, and caused by excessive toxic organic insecticides, and also helps in industrial sewage and also poisonous gases, biological control, and also poisonous gases. So now they are using the endophytes as a biological control to help to address these problems. And it found that it is an efficient method, and it become a widely used in killing insects or pathogens, and also this endophyte is also being used for environmental remediations. And also they have so many reports about endophytes being capable of producing antibiotics, which can be used instead of toxic organic compounds in preventing the beneficial microorganism spreading. So now they have some recent research has been done showing that these plant endophytes might be partially responsible for degradation of environmental toxins. The endophytes also is very interesting now. There are many research that has been, they use in the endophytes to degrade the biofilms. So this biofilm can be the contaminants in the environment. So they has the properties to degrade the biofilm by inhibiting the communication between bacterial communication in the biofilm. So this endophytes has the properties to quorum quenching properties to stop the communication between the bacteria. Hence, this endophytes has the ability to degrade the biofilms. The Methylobacterium populum is a novel bacteria, which is strain BGA001, existed as a plant endophytes, was involved in the degradation of energetic compounds. The Newman and Reynolds reported that the plants inoculated from the plants which is inoculated with VM1330, which is this one is an engineered endophyte, has been shown to increase plant tolerance to toluene and to decrease the transpiration of toluene to the atmosphere. So I'm about towards the end of my sharing sessions. So this endophytes has their this table shows that the endophytes and their produce natural product with special functions. Instead of being as an antibiotic producers, as anti-cancer, this endophytes also has the property to become immunosuppressant. They have immunosuppressive properties. For example, the endophyte fusarium subglutinans, which has been isolated from Tergium wilfordii. Another example, these endophytes can be an anti-diabetic agent. For example, the endophyte Pseudomassaria has the properties of the antibiotic agents. Endophyte cytonemia has properties as antiviral agents, and also there are so many other properties that yet to be discovered in endophytes. For example, they can be as pectin lysis they can have a pectin lysis activities, or they can have they can be as antioxidants.
Thank you, Dr. Izzah. So, any question from the audience? So, if anyone doesn't have any question, I think we can wrap up the session for today. And thank you, everyone, for staying with us. And let's meet again in the next session for another exciting talk from our Audience Fellow. And thank you. Till then, thank you everyone, and thank you to NCBI office for the opportunity.